Hi everyone, today I'm going to talk about something about trimethylamine nitrogen oxide derived zwitterionic polymers. Tri, tri means three, methyl means methyl groups, amine means this, oxide means this, so we call this trimethylamine nitrogen oxidase, which means TMOS. It's actually a small organic osmolyte present in sea waters. Yeah. To understand this, you can imagine something you learn in your high school textbook. When you put a cell in the salt water, it will contract because the difference of osmotic pressure between the cell and the solutions. So the cell will contract. To balance this, we need something to increase the osmotic pressure in the cells. So it is the T-mouse that act like this. To understand the properties of T-mouse, we need to know some terminologies. From the left side of the figure, it is non-folding, which means the protein will not adsorb on the surface of our materials. On the right is the biofoldings, which means proteins will adsorb on the surface of materials. Why? To understand this, we need to know something about zwitterionic materials. The Zwitter is actually a German. It means two. From the structure of TMOS, we actually can see there are two opposing charges, negative and positive. So it is actually a zwitterionic materials. The properties of zwitterionic materials is analog to water. Because it acts like water, so the protein will not recognize it and therefore will not adsorb on it. And for the material that is not zwitterionic, the protein will absorb it through protein-protein interactions, which mostly hydrophobic interactions. And also, the properties of non-folding of polyphenols is better than polycarboxybutane, which is also a zwitterionic materials from the butane. From the structure, we can see the opposing charge moieties of PMOS are directly connected with no space between two charges. However, for the carboxybutane, it contains at least one carbon separation. The longer carbon separation chain it has, the more hydrophobic it is. So the properties of non-folding will go down with the increasing of the carbon separation. To obtain the polymer, we need to do some chemicals. The starting material is not TMOS, it's actually dimethyl aminopropyl acrylamide, which is commercially available. And then we will oxidize it using hydrogen peroxide, which is a strong oxidized agent, which will result in completion of oxidation with no byproduct generation. And then after we get the TMOS, we will do the polymerization using ATRP, which is called atom transfer radical polymerization. To know the process, we need to know the mechanism of the ATRP. First, the initiator halide complex will be activated by the copper 1. And L in the figure means ligand, which helps to dissolve the copper salt in the solution. After activation, we will get initial radicals and it will react with monomers. However, the radicals will be deactivated by the copper 2, which will lower the concentration of radicals in the solution. It will maintain the rate of termination is low in the solution because the rate of deactivation is faster than the rate of activation. So the rate of termination in the solution is really low, which helps to make a narrow distribution of our polymers. However, if we use ATRP to do the polymerization process, we need to add a lot of copper salt in the solution because we want to maintain the concentration of copper salt really high because it will consume during the process. But the copper salt is not environmentally friendly. It will pollute the environment. So someone improved the process, which is called surface initiated ATRP. I say ATRP. In the process, we use copper plate instead of the copper salt solution. And the model is really like a sandwich. On the bottom, on the bottom is the copper plate, which is the catalyst. And in the middle is the filter paper with monomer solutions. On the top is the initiator modified substrate, such as glass or gold. After for about 1 hour, we will get a polymer brush, which is just like the toothbrush in your daily life. And the thickness of it is about 10 to 20 nanometers. We can actually measure it by using ellipsometry. Therefore, the advantages of the SI8RP is really obvious. First, it has a controllable thickness of polymer brush. By controlling the polymerization time, we can control the thickness of it. The more time we do the polymerization, the more thickness we will get. Also, it's really convenient to manipulate. First, we just need to dip our filter paper in the monomer solution and put it on the copper plate and cover it by the initiator substrate. And the process is really easy to manipulate, even for the beginners. After I sharing you these slides, I hope you can better understand the TMOS and how to make poly TMOS. Thank you.